Good afternoon. And for this relatively shorter session, I request Professor Arnab Bhattacharya from JNU to chair this session.
Thank you. Welcome this post-lunch session.
Yes.
And the first speaker is Professor Mrinal Srivastava from TFR Hyderabad, and she will speak on molecular programming of replication dynamics. So over to you.
Thank you very much. And good afternoon, everybody. And I hope you all had a good lunch. And I think it's great to have the first talk after the lunch, so there is no shortage of brain glucose to act on. So my name is Mrinal Srivastava. I'm from TFR Hyderabad. And today I'll be talking to you about molecular mechanisms of how cells decide from where to start replication, how to time, and how to maintain the patterns. And it's different for different cells. So in nutshell, my lab works on mechanism of replication, how cells maintains the fidelity of replication, and how our genome deals with different kind of adversities. So I will start from very, very basic. This is DNA replication. It's very, very fundamental process. When billions of years back, in the primordial soup of Earth, when Earth was just formed, the origin of life, the first criteria for origin of life is the system has to be self-replicating. The second is it has to have some kind of catalytic activity. It could be by RNA or DNA, but the first and foremost principle of anything which is living, that it has to divide its genome by its own machineries or by the help of someone else's machineries, like in viruses, then only they can be alive. Like this pointer cannot divide. This is non-living. But coronavirus, when it comes to our body, it can divide. It doesn't matter what kind of genome the organism has. It could be DNA as a nucleic acid or RNA genome. So because it originated that so many years back, there are certain things which are still remain same. That's the principle of biology, right? Biology is a tinkerer. We don't change machinery which doesn't need to be changed. If something is there, it's working perfectly, it's not need to be changed. But we tweak the system based on every organism we encounter. So if you see the replication patterns of single-stranded RNA viruses, very, very simple, versus bacteria or yeast or human beings, we come to the top of the food chain. The complexity increases. So the replication patterns, the molecular mechanisms which are important for DNA replication also change. But something remains same, right? We have every organism replicate by template guided manner. So, and it has two strands, leading and lagging strand, and direction of replication is conserved. 3' end has OH, new nucleotides are added to it. We don't have, from 3.7 billion years of evolution, we do not have a polymerase which can add to 5' phosphate. It doesn't have to be. One other thing which is conserved is semi-discontinuous. What does this mean is, leading strand is synthesized in a continuous fashion, and lagging strand, because the directionality has to be same, it divides in a small fragments called Okazaki fragments, and later they all get annealed or ligated. So this is something which is very, very conserved. But now I will say and go on and start telling you how we are not same as well. This is a very minimalistic expression or representation of eukaryotic cells, like let's say yeast. And I will tell you why I'm saying minimalistic. Because we are going to get into a complexer system, we have to see the minimalist machinery that's required. So let's say biomolecule, polymer, DNA in this case, is duplex. What's required is you need energy to break the bonds so they separate, because the information, the template, it comes from nucleotides, bases basically, and they are hidden. So they have to be exposed to some protein which can read it, polymerize a new chain. So MCM complex here, it's a helicase, it unwinds the DNA strands. And in eukaryotic system, we have 3 different major replicating polymerases: polymerase alpha for leading strand, polymerase delta for lagging strand, and polymerase alpha which synthesize RNA primers. RNA primers are required to initiate the synthesis, because again, our DNA polymerases cannot start synthesizing de novo manner. That means they cannot, from scratch with a pool of DNTPs, they cannot start adding 1 DNTPs at a time to make the polymer. And this also tells us that RNA world was before us. Because RNA genome can synthesize a primer, and we still are keeping that. We have not completely eliminated it. Because the genome becomes RNA to DNA, we got new DNA polymerases, but we did not remove, evolution did not remove something which is required to RNA polymerase. But that's a very minimalistic model, something to open up, something to polymerize. Inside our cells, mammalian cells, it does not really happen like that because we have metazoan, we have multicellularity, different parts of our body, like let's say liver cell versus kidney cells, our skin cells, they have different kind of gene expression patterns. So different kind of RNA is getting synthesized from different cells. And replication, when replication happens, all the marks have to be removed, all the histones have to be removed. And when new strand is made, cells have to be the same origin. Like if it's skins, let's say I get a wound here, and I get now the new cells have to be skin cells, right? They have to have expression of skin cells. They cannot just start expressing the liver cell enzymes or proteins. So first of all, that adds to the complexity because chromatin context and epigenome is different. And because RNA is also getting transcribed from different region, now ongoing replication machinery can collide with transcription machinery. Now it could be from behind or it could be in front. So there can be a collision, depends on what genomic region is getting transcribed in that particular cell. Our genome is very big. If we talk about diploid, 6.4 billion base pairs needs to be added in a short stretch of time to replicate in very high fidelity. High fidelity means if we calculate all the repair mechanisms, our polymerase specialization, it comes that it incorporates one error in 10 to the power 10 bases it reads from. Very, very high fidelity. But because we have such a big genome, instead of having a bDNA structure, which is most of us know it's a Watson-Crick bDNA form, but DNA can exist in different form depending on what kind of sequence context they have. It could be triplex, G quadruplex, R loops, they all have to be resolved before our machinery, which is specialized to read only B form of the DNA, can read from and replicate it from. I will talk about this in detail. And of course, in mammalian cells, we also have patterns, how the chromatin is actually organized. Something is transcribing a lot, something is transcribing very little, and we also have different kind of DNA damages. So it automatically tells you that something which is required to resolve this, probably different to resolve this. Like different kind of proteins needs to be. So basically, on this minimalistic model of replication, we need to have specific sets of proteins that deal with, let's say, G quadruplexes, or let's say a uracil kind of damage. So the replication machinery has a way of sensing what is it encountering, and something comes and specifically removes it. Because if we think about the DNA, when we go to cell signaling, checkpoint signaling or stress signaling, it looks very similar. It looks like no matter this is blocking replication fork or this is blocking replication fork, signaling is going to be same. But it's actually not, because the sensing mechanism has to be different. So why we are studying mammalian cells? Over the years, the first organism where people had studied DNA replication was a prokaryotic system. In fact, this is a system where people actually found out DNA is a molecule of inheritance. And a lot of our knowledge of replication, initially that DNA is semi-conservative replication, how it polymerizes, all these things comes from E. coli. But if we talk about eukaryotic replication, most of our knowledge, even till date, 80% of the people who work on DNA replication field, they work on yeast. The reason is the power of yeast genetics. It's unicellular organism. You can modulate and do very high quality of genetics, which is not really, mammalian cells are not amenable to it. And that's almost all these proteins, which I say on the minimalistic model, they all were discovered in yeast, and just by homology, they were transferred, sometimes without little evidence that this is how it happens in mammalian cells. If we talk about mammalian cell replication, we have a DNA virus genome, and that's the system people use to study mammalian replication. And recently, in vitro reconstitution and cryo-EM data has been really useful to understand how these giant machinery which are assembled and moving on the DNA works to replicate our genome. So the first question that we had asked is just that I introduced, that what happens if we have different kind of context. Is it going to be entirely different kind of proteins? Or like the way they can sense what kind of block is there in front of replication and what kind of context it is going through, versus it's a universal system that, let's say, replication folk is going to have a problem and specific set of proteins will come and they will elicit the signal. So how did we do that? What we did was we modeled two different kind of DNA damages. They both are cross-link damage. So either they cross-link two nucleotides in the DNA or they cross-link a protein to the DNA. So basically both of them are roadblocks. They're different kind of roadblocks. They either cross-link the DNA or they cross-link the protein. So we asked the question whether the replication machinery will sense just there's a block, or they are going to make a very distinction that this is a DNA-DNA crosslink versus a DNA-protein crosslink. So I don't want to go into detail with this study. We use a proteomic approach for both, which gives us a snapshot of the different time points versus which gives us a process. And at the end, we found that if you have DNA-protein crosslink or UV crosslink, which is a DNA-DNA crosslink, you get entirely different sets of proteins. Although the cell signaling machinery downstream is same, cells have a way to recognize what kind of damage it is so that they can resolve it and move on. So they are so this is the only example that we have for DNA damage. So it also tells in extrapolation that if you have different non-BDNA structure or transcription replication conflict versus DNA, different kind of chromatin context, it's going to have a different set of protein machineries. Okay, so coming to as I said, I will be talking about a little bit more about replication timing and regulation of origin firing. This is very, very specific to mammalian cell issue. So for that, we will talk about where, when, and how mammalian replication starts. Why this is a very specific problem with mammalian replication. The reason is when we think about bacteria, they have one origin of replication. That is the one from where replication initiates. And when we talk about yeast, although they have more than one origin, but we still know the sequence and we know the frequency of their firing. So we exactly, even in yeast system, know from where replication is going to start. And when we come to mammalian cells, we don't know absolutely from anywhere replication can start. And there have been a lot of sequencing efforts of different ways, and the patterns doesn't really give any answer at the end. The reason is mammalian cells have a lot of replication of origin sites. So let's say these all blue dots are origin of replication from where replication can initiate. Basically focus on the green dot. I don't want to talk about the active constitutive origin. That means they always fire. They have higher chance of firing at any cell cycle. So out of these small dots, any one of them can fire. What all you also have to notice is if this one fires, origin just simply next to it does not fire. The reason is our replication fork speed is somewhere around, let's say, 2000 to 4000 nucleotide per minute, which is quite remarkable. So the replication machine somehow knows that it can this one doesn't need to fire for it to replicate. The far away region needs to be fired, but this one doesn't need to be fired. So they have a way by which they maintain the distance, how replication origins are going to fire. But why do we need all these origins? So you will see in my later slides, mammalian cells, if we collate all the replication sequencing data, which people have put a lot of effort into finding where mammalian replication starts, is somewhere around 350,000 to 550,000. So those are the regions from where mammalian replication potentially can start. But out of those 500,000 potential origin, we actually only use 20,000 to 30,000 origins. That is like only 5–10% we use. 90% of them are never used in the cells. And the lot of effort goes into licensing a site. Licensing a site means you put certain proteins to mark this is my origin of replication, and you never use it. So why do cells even do this? The reason is, let's say something happens. This is the origin which is going bidirectionally, symmetrically, but one side of it has some kind of stall. Now what happens? The downstream one, which in general scenario would not fire because it doesn't need to fire. It's moving bidirectionally. Replication folks somehow knows it doesn't need to fire downstream one or the nearest upstream one. But now on the other one side, because there's a block, the downstream one can fire. This is important because so that we do not have any problem or genetic mutations incorporated while replication. So the because it's a fundamental principle of the origin of life that we need to, with very high fidelity and very high care, need to replicate our genome. And that's the benefit of having so many flexible origin of replications. So over the years, there have been a lot of thought has gone into it, that how mammalian cells decide which origin to make. Is the frequency same? Does the frequency increases? Is maybe there's a domino like of model, one place randomly it starts, then it's just a domino effect, and it keeps going. And every model has certain flaw. Like for example, I'm just giving two examples here. In one model, they say the frequency of is just a, let's say, hypothetical model. There are 100 origins here, and the firing percentage or frequency of firing of origin remains same. In certain scenario, let's say nothing fires in the beginning on the right side of this cartoon, then you will have a gap problem. It takes really long time to replicate completely, right? And our genome, this is only 100, very small stretch. 6.4 billion base pairs have to be incorporated. If it will become very long replication. So this problem can get solved if you think the frequency of firing increases. And this can be explained by, if you explain there's a limiting factor in the cells, which slowly gets more and more, because so cells do not replicate. So every time this empty circle becomes red circle, whatever protein was licensing that as a origin replication is displaced. So that ensures that once replicated, it will not re-replicate in that cell cycle. So that's why the limiting factor concentration is increasing, the frequency is increasing, your replication time is less. The problem with this model, even dominant model or any other, there are a lot of models, is the mammalian replication is not entirely stochastic. That means we can divide our genome into certain replication domains. So if you divide S phase and mid, early and late for a particular cell type, let's say we're talking about skin cells, let's say for skin cell, regions of genome that replicate early, they will replicate early. Regions that replicate in mid phase, they will replicate mid, and regions that replicate late, they will replicate late. So that's not, so that says there's no, not, it's not completely chaotic. Yes, no. So in one chromosome, we can have alternating early, mid, and late. So that cannot explain. Yeah, no. So like one chromosome itself, in this, like this small portion itself, you can see something is replicating late, something is replicating early. So it's not, but what we know is in one cell type, this pattern is going to be same. But within that region, there will be multiple origins. Which one of them fire is totally random. Yes, maybe 10 to 20%. Yes, that's very good question. So actually, if we, let's say a normal cell culture, HeLa cells. So every HeLa cell will use different origins. That's the reason sequencing did not give us much. The only thing what we get to know is within that domain, let's say something is replicating early, in that it can use any origin. But that region of the genome will always replicate early. So there is randomness, but not entirely. Yes.
Can I make a comment here?
Yes.
I mean, a theoretician collaborator of Rind, John Beckhofer, maybe you know already this. He, of course, developed a theory,
yes,
which is, this is a nucleation and a growth problem. So all these potential sites from where this can start, yes, so they nucleate, but of course it's a random process, so they will not nucleate simultaneously.
Yes.
And once they fire, then after the actual replication fork that propagates was mapped onto a growth problem. So it was converted into nucleation and growth problem, and which is a stochastic process theory.
Yes. So it is a stochastic process for defining which origin fire, but it's not entirely stochastic. Because as I was talking to Sandhya's answer, if we look at this arm of the chromosome, only this region itself, you can see early and late. So there's a boundary.
So there may be some barrier which has to be crossed, and so when that, it takes its time depending on the barrier. So
yes. No, at different, it also.
Anyway, okay, we can come back to this question later.
So this is just to summarize what I said. You have same cell type and different origin can fire. And it also has a sensor. So let's say this one fires, we know the next one is supposed to remain dormant, but something happens to it, then downstream will start firing. If otherwise, it will not fire. So they have a zone of inhibition and zone of activation. So these are the factors that we can think of how replication patterns are decided. First, number of potential origins. So what is the frequency of, if you reduce the number of origins, of course, the more origins have to be fired. And if they are not uniformly distributed versus early, late, and mid, then we will have a different patterns. Fork speed and symmetry. Fork speed also is not same, depending on euchromatin, heterochromatin context, whether there is a DNA damage or not, the fork speed can go higher or fork speed can go low. So what does that mean is we have molecular regulators, we regulate the speed of the fork as well. Distance parameter of induced firing, we have seen with the random gap problem. So we do not need lot of the origins firing, but let's say there is no origin firing till 100 or 200 or 300 kb region or more, then you will have more problem because we expect one replication fork which started 200 kb or 300 kb downstream to processively completely go. Then we have that kind of issue. And of course, we don't want to fire an origin which is, let's say, close by 10, 20, 30 kb as well. So there's a distance parameter of firing inhibition. And of course, firing probability in euchromatin and heterochromatin is different. And this is not exhaustive list. This is the thing that we thought, because we want to test it experimentally, which is very, very difficult. So we thought, let's test some of them, which we can modulate on experimental level, because mammalian cells are not very amenable to having removed a replication protein or overexpressed a replication protein. Both becomes a lethal after a cell cycle. So we knew that we cannot knock down a protein, knock out a protein, overexpress a protein. Then how do we actually study this? And that's the reason, because we don't know how replicons are regulated, we absolutely have no idea how cells make when some origin fires, downstream remains dormant but can be reawakened again if there's a block or some kind of replication stress. So we thought that maybe we can modulate some of these parameters. Maybe we can reduce the number of potential origins. Maybe we can try to modulate the fork speed and symmetry. So if it's asymmetric, maybe one side will have more origin firing versus less origin firing. So how do we do it? This I already mentioned that this is what we wanted to do. So this is a very complicated process of replication, licensing, activation, firing. I don't want to go in entire detail of this. I already said there are proteins which, when bind to DNA, mark the sites of origins. And mammalian cells, it could be 350,000 to 500,000, depending on which sequencing paper we are looking at. This particular process happens only in G1. So if you see all the origins, this is the MCM helicase, which gets loaded only in G1. Up to this point, everything is same. By proteomic wise, we do not know absolutely any difference. Everything is same. What happens is, when cells enter in S phase, some of these origins, something else is happening here, which is not happening in nearby origins. And it could be totally random. But what we have to see is, if this is something happening by phosphorylation, then phosphorylation should happen in all of them, because the CDK, DDK levels in S phase, they increase as cells enter to S phase. So all of MCM should get fired. Because kinase, if you see the kinetics is very, very fast, but it does not. Now we can say there's a opposing phosphatase activity, but that will not explain, because again, I already mentioned, it's not entirely random, because the early one will be replicating early, late one will be replicating late for a given cell type. But definitely something is happening, and this origin, something is happening here, which is getting activated, it gives some signal to the downstream close by origins, don't fire unless I get into some kind of problem. If I'm getting into a problem, then you have to fire. And once it reaches up to here, it will displace it. So this will not be site of origin anymore if it successfully replicates through that process. So what I want you to understand that this step is called licensing. It happens in G1 phase, which marks the site of origin. And something happens here, which is in one of the origin, which is activation. And this step is called firing, when we actually see replication going bidirectionally. So we thought, let's modulate something on the proteomic level at these three distinct states. If we do this here, maybe we'll have less of the origin, less of the activation or poor helicase translocation, and less of the firing. So what will happen? So the idea here is, let's say this helicase is moving, but its helicase is moving slow. Would the cells recognize now this one has to fire, because the time going to be taken between these two origins will be more. So these are the three steps that we thought we can modulate, and we are doing it using, so before I'll go ahead, I will mention that a lot of biochemistry has gone into it. So we identified, and we also took, of course, a lot of help came from cryo-EM structure-guided mutations, and a very, very rare mutation that humans have, which is called Meyer-Gorlin syndrome. And in that, some of the proteins related to replication forks are mutated, which is very rare because we don't really see a lot of mutations in replication cycle, not even in cancer and any other genetic disorder, because if you have mutation in co-replication proteins, you are not probably going to see an organism. So it's an extremely rare genetic disorder. And we have done some biochemistry, and then those mutations we have modeled into certain proteins like CDT1 here, which is basically a helicase loader. And this is a CDC45, which comes only at the activation step. This protein is only present. Up to this step, everything is same. But this step, there's a protein called CDC45, which binds only to activated MCM helicase. And what people think is, it basically guards the gate. So DNA has to be in the center of it, and it guards the gate of MCM225, CDC45. And if you have not guarding the gate properly, what happens is DNA can actually fall out. So speed of helicase will tremendously decrease. And then we have a factor called MCM10, which actually recruits polymerase alpha, also increase the unwinding activity of MCM helicase to basically make it translocating, because we know that RNA primer has to be there in order to replication to happen. And as I already mentioned, all these proteins, they get regulated very tightly in the cells. The cell cycle actually, G1 to S transition entire is based on how replication is happening. In fact, G2M also, whether replication is complete or not. So all these proteins gets regulated not only by expression levels. Of course, we don't have CDT1 in S phase, because if we have CDT1 in S phase, it will load MCM protein again in the replicated genome, and we will have ploidy chain. Instead of diploid genome, we will have polyploid genome. So that's why CDT1 is not free to load MCM complex in G1. What I'm trying to tell you is, if we want to model these mutations, we cannot just overexpress it from a random plasmid and model these mutations. Otherwise, that would be very, very easy. So what we did was, we modeled these are point mutations, different mutations in CDT1, MCM10. We are still working on CDC45 mutations inside the cells and the endogenous locus. Because these are point mutation, we cannot see whether the point mutation has happened or not by PCR, simply because the genome size is going to be, the fragment length is going to be same. So what we have done is, along with the mutation, we have also incorporated a novel restriction silent site, and then we can screen. So I see this one gel, but I know one of my students, Muntaj, how much work it takes to get to this final gel. So we are still working on CDC45, and hopefully we can modulate some of these parameters and eliminate a very, very small region of the mammalian replication. Because we even know the experimental challenge, this is very challenging, but after this, there's another level of experimental challenge that we cannot actually study entire mid-early phase and which origin is firing. We can look at the global level, but if we look at a small domain, it's very limited regions we can look at. We can look at 3 to 5 places. So as I already mentioned, another way to modulate the program of replication is expressing an oncogene. Cancer cells do it all the time. They divide faster, divides uninhibited manner, and lot of origins in replicate. If you overexpress simply an oncogene, you can see the origins that never fire in that particular cell line will start firing. So a lot of close by origins will start firing. And another thing oncogenes do they basically make replication asymmetric. So this is what we are also trying to model in primary fibroblasts and see what's happening to the neighboring downstream origins. So we are still working on this. So this is the ongoing studies for this part of the project, that we are trying to see how under-licensing or reducing the translocation speed of MCM complex is going to affect the origin firing. And because as I said, we can only illuminate, we can get experimental data for only very few regions. So if we want to extrapolate into different regions, we of course need to model it, but with more certainty now. It's not replication models are not done, but they have been done on assumption because there was no information of replicon regulation. So second part of my talk is about what happens. So everything is going, still now what we were looking at, everything was normal in normal replication. But what happens when replication fork, let's say this is the replication fork, encounters any kind of block. This is a very big block, it could be a very small block. Because our replicative polymerases, I already mentioned, they rarely make an error. Through the evolution, they have been perfected that they can only incorporate correct nucleotides against the bases. So when we have something abnormal, they do not know what to do most of the time. Otherwise, they will be incorporating something wrong. So, but replication fork cannot be stopped. In the cell cycle, in the G1 phase, there's a point called restriction point. Once the cell crosses that point, they have to divide. They have to duplicate their entire genome. So if replication goes halfway, either you're going to have genomic instability problem or cells are going to be dead. There's no going back after that point. So replication, generally cells is given maximum priority and replication is generally never stopped. So what happens is, again, I don't want to go into a lot of detail in the process. Let's say this is polymerase delta, which is a replicative polymerase of lagging strand, and this two yellow, red nucleotides here with bases, basically this is the abnormal bases. So polymerase delta is stuck here. What happens here is there's another polymerase which is called translesion polymerase, as its name suggests, is just basically polymerized translesion. There's no repair happening here. But its active site is bigger than replicative polymerase, and we have 13 of different polymerases other than replicative polymerases in human cells, which is a lot than any other organism. So this translesion polymerase, it could be polymerase eta here, but as I said, it could be any 5, 6, 7 of the other polymerases. So polymerase delta is evicted out of the replication fork. Polymerase eta is incorporated. Polymerase eta can incorporate right or wrong, depending on what kind of damage it has, what kind of bias it has towards that. This is called lesion bypass. This lesion is still there. Only thing is some nucleotides are added against it because otherwise replicative polymerase was stuck here. It had no clue what to add against it. So after that, polymerase eta has bypassed this. And once it bypasses, because it's error prone, it's going to generate mutations. There's another mechanism by which polymerase eta is evicted and polymerase delta is put back. So these processes are called polymerase switches. This is one polymerase switch, the second polymerase switch. Okay. So this is the questions that we still have in the field, that how these polymerase switch happens. We have experimental data to see the residence time of each translation polymerase. So we know that they don't stay for very long. What we do not know is what recruits them and what removes them. So how this polymerase switch happens, the first one as well as the second one. What are the changes that happens on the replication fork? Although this is only showing the clamp, I showed you the replication machinery. What kind of changes that happens on the replication fork when they encounter any error-prone polymerase? And as I said, there are a lot of TLS polymerases in the cells, so which polymerase comes when and how, that's also not known. And we do not know if the replisome change that happens in polymerase eta is going to be same as, let's say, some other polymerase, translation polymerase comes, or it's going to be different. This also can be totally stochastic. Maybe the TLS polymerase which is close by, that gets recruited and job is done. But if that would have been the case, why cells would not invest on just increasing the levels of translation polymerase instead of making so many different family of translation polymerases? So these are the questions that we have. So in one of the earlier studies that we had done, we had identified a protein called HMCES, and this protein we could only find on the replisome machinery when there was a bulky adduct present in front of replication fork. This is a classical example of UV damage. You can see cyclone pyrimidine dimer or pyrimidine pyrimidone dimer. Those both, basically what they do is they stall replication fork. So what we did not know what is the role of this protein, and so we did a lot of different assays. We found that it interacts with pCNA directly and that's how it gets loaded. But what does it do to translation synthesis? Because specific to these kind of adducts. So what we did was, so now we wanted to have a damage in front of replication fork, not somewhere away from replication fork, which was also a bit difficult. So what we did was we used a metabolite, which increases the level of uracil in the cells, and it can be incorporated in the DNA only in a replication dependent manner. So replication is machinery itself is going to incorporate it, and that's how it's going to encounter it as well, because uracil cannot go into genome on its own. What we found is, when we have control cells, this is anti-metabolite, which is basically used in lung cancer, a lot of other, it's a first-line chemotherapy these days. We have control cell in 1 micromolar, all the cells you can see almost dead. But our HMCS knockout cells, they're very happily alive. Now look at this one, we even increased it further. Everything is dead by here. But if you see closely, even in this knockout cells, there's still some cells there, which is very surprising. You can remove a protein and cells can survive at very high concentration where wild types cannot survive. So we did a lot of other studies, and this was true with 5-fluorouracil as well. We did a lot of studies and we found out that this HMCS somehow regulates the translation synthesis pathway. And of course, there's a because translation synthesis pathway, basically if it's going to be overutilized, it's going to increase the mutation. So what is going to happen here? Let's say there's no transition polymerase. Polymerase delta sits here, does not have any clue what to do here. It's going to stall. When replication stalls for a long time, I've mentioned cells will die. But because of polymerase eta here, it can bypass the lesion and cells can happily live. And that's what we were seeing here. When in control cells, transition synthesis is very well regulated. So our levels of transition synthesis, so our cells are dead when you give that much, they have that much mutation. But when you remove HMCES, cells can survive. So that means these cells should also have higher level of mutations. And that's what we found as well, that HMCS cells, they have higher levels of mutations. Somehow HMCS regulates a transition polymerase, but this was only true with, I am not showing that data, but it was only true with polymerase eta. It was not regulating any other translation synthesis polymerase. That also tells us that each translation polymerase regulation, its recruitment and resolution depends on different sets of protein. It's not a universal mechanism. Yes. I don't think people have really looked into it. So I don't think people have looked translation synthesis utilization in early, mid, and late. Even in fact, in our studies also, we are just looking at S phase at its entirety. Yeah.
But since you know the regions, couldn't you now go back and look? Because I thought there might be a preference for certain regions which maybe are more sensitive or go faster.
Yes.
To accumulate more.
Yes. So accumulation of damage is all that bias we know. We do not know the bias of translation synthesis. So genomes that are region to open, if you have UV treatment, they will get more damaged as well. Yeah, that's a good point. We can look into it, that translation synthesis utilization in early, late, and mid phase of the cell cycle. Yes. So as I was saying that this was only true, HMCS regulation of translation polymerase was only true in case of polymerase eta. So that told us, it did not tell us the mystery of translation polymerase regulation, but it told us that every TLS polymerase gets regulated in a different manner. So there is no easy way out. If you want to study translation polymerase, you need to study one by one. That's what information we got from here. So one of my students, Snigdha, what she's trying to do, she's trying to basically knock in. So we have two questions here. Let's say we have a damage, any kind of damage A. As I said, there are different types of, there's an entire class of alternative polymerases that can be utilized. We want to know which one comes, if there is a bias, depending on what kind of damage it has, and how fast they go away. Or maybe it's not one TLS polymerase that comes, it's also possible that two TLS polymerase comes, one inserts and other one extends. So we want to first see the kinetics and resolution of different TLS polymerases. And second, of course, we want to understand the mechanism of TLS polymerase switch, the polymerase switch that we saw. So what my student Snigdha is doing it, she's basically trying to incorporate, this is one of the example which she recently did was, this is last exon of polymerase alpha, which is so there are 3 replicated polymerases also. So the permutation combinations are more. Let's say polymerase alpha has to be removed and TLS has to be put. polymer delta has to be removed, T-less polymerase has to be put, and polymer alpha has to be removed and T-less polymerase have to be put. And then T-less polymerases are many, many. So there are different levels of challenges here. So she is trying to make these mutations in 3 of the replicative polymerases because they have to be switched back and forth, and couple of chosen T-less polymerases to see what is the proteomic change that's happening. So what she is trying to do here is, as you can see, there's supposed to be a stop codon here. The stop codon is removed, and this is the proximity labeling bio-ID tag that has been incorporated in the genomic locus. Along with that, she's also trying to incorporate a split fast tag. How does this work is, you have, basically it's a fast tag which has been split. One of the portion of the split fast tag you put on the replication machinery, like shown here. This particular circle is called clamp, which is very, very important for replication. It basically holds or tethers the replicative polymerases. People have shown that it also makes contact with T-less polymerases. So utilizing that information, 1/2 of the split fast tag has been put into the clamp, and other part of the fast tag has been put on the polymerase eta. So they will not fluoresce if they are separated, but when they are together and you put a ligand, they will start to fluoresce. And this we are going to do it in different T-less polymerases as well. But the problem here is, again, a lot of biochemistry has gone into it, because you need to understand which region comes close to clamp. Just because putting an N-terminal, C-terminal randomly, it's not going to help us, because it's not going to give you a proper fusion protein. You probably have to put a linker. So before we went ahead and did, you can understand, before we do one experiment, we have to do at least 10, 20 experiments before that to figure out which region of each we need to put actually these split fast. But some of the polymerases we are still working on, some of the polymerases we already had some success. Based on that information, we have tagged all 3 replicative polymerases. This is just a confirmation for polymerase alpha and epsilon. And Snigdha is in process to tag polymerase, T-less polymerases as well. So another thing that has been incorporated along with the tag is something called FKBP and FRBs. What they do is, so one way to look at them, let them, whichever polymerase comes, let them come and let them go away, depending on damage. Other thing is you can force recruit them, because one information that is still missing in the field is what kind of mutation profile these T-less polymerases do and why it is important. In lower eukaryotes like E. coli, translation synthesis is considered a major evolutionary force. One of the major ways we incorporate mutations. Mutations are bad, but not all mutations are bad. Like coronavirus mutations. For coronavirus, some of them are good, most of them might be bad, or most of the mutations are bad, but for evolution, they are very, very important. And because we do not know T-less polymerase mutation profile, we do not know naturally what kind of mutation profiles they do. We do know in cancer cells there are a lot of mutations, and we know T-less translation synthesis machinery is one of the major mutation incorporating machinery. So if we have that information, probably we can look at the cancer genome and say what kind of translation synthesis machinery is being used, because it can almost bypass all the bulky lesions. We use a lot of cancer therapy drugs rely on incorporating damages to the DNA, like cisplatin, or I already mentioned pembrolizumab 5FU, almost all of them. So this is the second thing that we are trying to do, along with studying the kinetics and recruitment and proteomics. We also want to do what is the mutation spectrum analysis of each polymerase recruitment, if we forcefully do that. So for this part, we are trying to study whether this translation polymerase, each translation polymerase work through a specific mechanism which recruits them and resolve them, or it's a similar one, which our previous studies have already ruled out there's a universal mechanism for all T-less polymerases. And there's a preference for particular translation polymerase over the other, depending on the damage. So if there's a specific kind of damage, that particular T-less polymerase is on purpose recruited. It's just not stochastic that happened to be close by and recruited. So that's another thing that we want to see. So with that, I would also like to thank our collaborators. We have some great collaboration with physician scientists from CDFD and PGI Chandigarh. So that's how we are getting our patient fibroblasts. And we're also getting some very rare sample, thanks to CDFD, from this Meyer-Gorlin syndrome, because it's one of the rare syndrome. It's so rare that we don't even know its frequency. So it really happens very rarely, but they do have some precious samples of those patients, and we can look into that. And for the modeling, we are taking help with Dr. Anupam Gupta from Department of Physics in IIT Hyderabad. So this is my lab. I should talk to you about work of Muntash and Snigdha. Manjul is working on single molecule study of clamp translocation and the nuclease. And Rajani, she's working on oncogene-induced replication stress. All my students are very, very new. They just joined 6 to 8 months back, but they have done some excellent work. And of course, I want to thank TFR internal intramural funding and also all my colleagues in TFR Hyderabad, because I joined, had nothing, but we could start our work without any delay, thanks to them. And of course, I want to thank DBT. I have a grant from DBT as well. Thank you, and I'll be very happy to take questions.
We have time for questions. Any question?
Yeah, I have a question. Whenever you have talked about polymerase, it was always DNA polymerase, right?
Yes.
Yeah, but same DNA stretch may be used also simultaneously for transcription also.
RNA polymerase.
So do you have, I mean, I know it's too early. Do you have plans to look at the interplay of the two different polymerases using the same track?
That's a fantastic question, because RNA polymerase also gets loaded back to back. And like DNA polymerase, the chances of collision in active region is actually very, very high. And this is an active area of research also. So people think there are different kind of machinery involved depending on it's a head-on collision or both of them are going back to back and speed difference is there. But exact molecular mechanism, I think, is still not completely cured. Maybe we will look into it, because
Yeah, see, being theorists, we can of course always can study such. We did that, but we found at that time that there are two competing possibilities. One was that the DNA can switch over from the one strand to another, and another can bypass, and so on and so forth. So whether this bypassing actually happens or not when they have head-on collision, so that is something. There are very few experimental results. Maybe
yeah,
worth
actually,
looking.
that's interesting, because that same thing with transition synthesis also. We think that, let's say, because RNA polymerase is going to bind only one strand, but DNA polymerases are going to bind two strands. But the lagging strand, if lagging strand collides with RNA polymerase, that's very simple. It can just make a Okazaki fragment after that and replicate. That's what people think with lagging strand also. But there is a limit there also. So lagging strand, it's not like always is bypassed, because leading and lagging strand, they're coupled. It's not like leading strand can go on without lagging strand or vice versa. So it can happen, but I think it also depends on how much distance it is. I think experimentally it has to be
Exactly.
validated.
Yeah. Okay, thank you.
Other questions?
Yes.
When you talked about DNA polymerase, so have you depleted, as you said that we cannot remove DNA polymerases or overexpress it because it will be lethal. So have you tried, there are groups, so down regulating a few of them and then checking that how does it affect the.
So this DNA replicated polymerases we cannot knock out, but translation polymerases we can knock out. They are not lethal. Only the core replication machinery is difficult. So for different TRS polymerases, if we knock out, let's say, transition synthesis, people have shown little mutation increase, sometimes no mutation increase, depending on the DNA damage they use. Sensitivity of the cells toward that damage is increased.
But that will not affect the origin of replication.
Yes.
Okay. Any more question? Okay, so I just want to understand this. The polymerase, the choice of polymerase, is it it is not obviously stochastic, right? So
that's what we want to explore.
Yeah. So depending on what kind of problem is there,
yes,
it selects. But does that impart some sort of changes in the machinery, replication machinery, so that it can actually select
recruit something? That's what we think might be happening, but we exactly do not know what kind of exact change that's happening there. So people know that PCNA gets ubiquitinated, but that's just one ubiquitination will not select between the TLS polymerases. So we do think that some specific kind of modulations are happening there, but we do not yet have any idea that what is specific for what polymerase.
Okay. Okay. So if there are some more questions, let us thank the speaker once again.
Thank you very much.